Welcome to the deep dive. Get this, a high-quality genome study of a Neanderthal woman from Siberia. Well, it showed her parents were as closely related as half-siblings.
Yeah, I mean, think about that.
It really suggests that level of inbreeding wasn't necessarily a super rare thing in her recent family history.
Exactly. And it really shakes up those simpler, older ideas about human evolution being just a straight line, doesn't it?
Totally. It points to a much more complex, interwoven history with different human groups actually, well, meeting and mixing.
That's right. Because before this 2013 paper, I mean, our genetic picture of Neanderthals was fuzzy. We had bits and pieces, low coverage genomes.
Right, like that earlier 1.3-fold coverage one.
Yeah, and even the Denisovan genome, though much better at 30-fold, didn't give us the full picture from Neanderthals. With those earlier drafts, it was just hard to accurately measure things like genetic diversity or spot close relationships.
Or track gene flow properly.
Precisely. It was like trying to understand a complex story from just a few blurry photos. So this research, the goal was really ambitious. Get a Neanderthal genome with modern human-level accuracy.
To tackle those big questions head-on, about interbreeding between Neanderthals, Denisovans, maybe even older groups.
Exactly that.
Okay. So the mission for us in this deep dive is to really get into this landmark 2013 Nature paper, the one that gave us this amazing complete genome of the Neanderthal woman from the Altai Mountains in Siberia.
We'll look at the findings, obviously, but also some of the clever methods they had to use.
Yeah, and why it's just had such a massive impact. We might even touch on, some of the general challenges researchers face when they're digging into these dense scientific papers.
Sounds good. Let's start with the physical evidence, then. Figure 1 in the paper. It shows the key find, this toe bone fragment, a phalanx.
Found in Denisova Cave, right? Back in 2010.
Yes, and Denisova Cave is it's such a critical site because it's the only place we found physical remains of both Neanderthals and Denisovans.
In the same rough time period, even the same layer.
Pretty much. This toe bone and that famous Denisovan finger bone came from the same main archaeological layer, layer 11, which is dated to at least 50,000 years old.
And there were sublayers mentioned.
The finger bone was in sublayer 11.2, dated to about 50,300 years ago, plus or minus a couple thousand. The toe bone was actually from a slightly lower sublayer, 11.4.
So potentially a bit older.
Potentially, yes. And interestingly, the shape of the toe bone, its morphology, it had features that reminded people of both Neanderthals and modern humans. So initially, it wasn't immediately obvious what it was just by looking.
Made the genetics even more crucial. And figure 1 also shows the other key Neanderthal sites with genome data.
That's right. It gives you the geographical context. You've got Vindija Cave in Croatia, Mezmatskaya Cave in the Caucasus, and then Denisova Cave way over in the Altai Mountains of Siberia. Sets the stage.
Okay, so the toe bone is piece 1. Then comes the genetics. What did that tell them? Figure 2, right?
Figure 2 is where the story really unfolds genetically. They start with figure 2a, looking at the mitochondrial DNA.
That's the smaller piece of DNA, passed down only from the mother.
Exactly. And the analysis showed the Altai toes mtDNA grouping really tightly with other known Neanderthal mtDNAs. They formed their own distinct branch, separate from modern humans, and, importantly, separate from the Denisovan mtDNA.
Any specific Neanderthal was closest to?
Yes, its closest relative among the known samples was the mtDNA from a Neanderthal infant found in Mezmatskaya Cave, the one in the Caucasus.
So a strong early clue this was a Neanderthal.
A very strong clue. But the real confirmation comes from the main genome, the nuclear DNA. That's figure 2b.
Okay, what did the nuclear DNA analysis show? They used transversions, it says.
Yeah, transversions are a specific type of DNA-based change, like A switching to T or G switching to C. They're often used in deeper evolutionary comparisons. And using those, the analysis placed the Altai individual firmly within the Neanderthal evolutionary branch.
Closer to other Neanderthals than to the Denisovan.
Definitely. Genetically closer to the Neanderthals from Vindija and Mezmatskaya than to the Denisovan individual found in the same cave. They even quantified it.
How different was she?
The sequence divergence, the amount of genetic difference between her and the other Neanderthals, was only about a third of the divergence between Neanderthals as a group and Denisovans.
Okay, so that seals it. This toe bone belonged to a Neanderthal. The Altai Neanderthal, as she's now known.
Precisely.
The paper mentions using a neighbor joining tree and bootstrap values. Can you quickly unpack what those mean for us?
Sure. So think of the neighbor joining tree, like the one in figure 2b, as basically a map showing genetic similarity. individuals or groups that are genetically closer clustered together on the branches.
Makes sense. And the bootstrap values.
Right. Well, ancient DNA can be tricky, sometimes damaged, so you want to be sure the branches on your tree are, statistically robust, not just artifacts. Bootstrapping is a way to check that.
How does it work?
You essentially resample your genetic data many, many times and rebuild the tree each time. The bootstrap value on a specific branch tells you what percentage of those times that same grouping, that same branch appeared.
Ah, so high values mean more confidence.
Exactly. Values over, say, 70 or 80% generally give you pretty good confidence that the relationship shown on that branch is real and well supported by the data. The values on the key branches in Figure 2b were high.
Got it. So she's definitely a Neanderthal from the Altai Mountains. Now, let's get to the juicy bit. The inbreeding. How on earth did they figure that out?
The key was looking for something called runs of homozygosity in her genome.
Homozygosity, meaning identical genetic information.
Right. Long stretches along the chromosomes where the DNA inherited from her mother was identical to the DNA inherited from her father. We all have some, but what was really striking here was the amount and the length of these runs in the Altai Neanderthal genome.
Compared to others.
Significantly more and significantly longer runs compared to what they saw in the Denisovan genome, for example, or in most modern humans.
And why do long runs mean inbreeding?
Because if your parents are closely related, there's a much higher chance they inherited identical segments of DNA from their recent common ancestor or ancestors. And they're more likely to pass those identical segments down to you. The longer the identical segment, the more recent the common ancestor likely was.
Okay. And they could actually estimate how closely related her parents were. This is where the half-sibling thing comes in.
Exactly. They ran computer simulations to see what level of relatedness between parents would most likely produce the pattern of homozygosity they observed. The best fit suggested an inbreeding coefficient of 18.
Which translates to?
That translates to the level of relatedness you'd expect if the parents were half-siblings. But the paper is careful to point out other possibilities.
Like what?
Relationships like double first cousins, or an uncle-niece or aunt-nephew pairing, or even grandparent-grandchild could potentially result in a similar amount of homozygosity. Figure 3b in the paper actually diagrams some of these potential family trees.
Did anything else help narrow it down?
Yes, finding long homozygous regions on the X chromosome was also informative. Because of how X chromosomes are inherited, finding those helped rule out certain parental relationship scenarios that wouldn't typically produce long runs of homozygosity on the X.
Wow. That's quite a specific insight into one individual's family. Was this just like a one-off situation for her family, or did it hint at something broader about the population she lived in?
That's the next really interesting question they looked at using Figure 3c, even after they sort of mathematically accounted for that very recent half-sibling level inbreeding event.
Right.
The Altai Neanderthal still showed more runs of homozygosity, particularly in the medium-length range, than the Denisovan genome did, and even more than a present-day human population, the Caritiana, who are known to have a relatively small population size and thus lower genetic diversity.
So what does that suggest?
It hints that maybe small population sizes and consequently a higher background level of mating between relatives might have been relatively common within this specific Altai Neanderthal population, not just in this one instance.
But they need more data to be sure if this is typical for all Neanderthals.
Absolutely. This is one high-quality genome from one individual in one specific place and time. We need more high-quality genomes from different Neanderthal groups across their range to see if this pattern holds elsewhere or if the Altai group was perhaps particularly isolated.
Makes sense. Okay, shifting gears slightly. The study also estimated when these different ancient human groups might have split from each other evolutionarily. That's table 1, right? Population split times.
Yes. Using the high-quality genome data allows for more refined estimates of these deep divergence times. They used a couple of different analytical approaches.
Like PSMC, what's that?
PSMC stands for Pairwise Sequentially Markovian Coalescent Model. It's a method that looks at the patterns of genetic variation within a single genome to infer past population size changes and split times. They also used a method comparing allele frequencies between different populations.
What did they find? When did our lineage split from the Neanderthal Denisovan line?
Based on their analyses, they estimated that split happened somewhere in the range of 553,000 to 765,000 years ago.
And the split between Neanderthals and Denisovans themselves.
That was estimated to be more recent, around 381,000 to 473,000 years ago.
Those seem like fairly wide ranges.
They are, and it's really important to remember these are estimates. A lot depends on the assumptions you make, particularly about the mutation rate, how quickly DNA changes over time. They actually use two different calibrations for the human mutation rate, which is part of why you get those ranges.
So useful timelines, but not exact dates set in stone.
Precisely. The models are simplifications of what was likely a very complex demographic history, but they give us a solid framework.
Okay, so we have the splits. What about the mixing, the gene flow? This study was huge for pinning down the Neanderthal contribution to modern humans, wasn't it?
Absolutely crucial. Having this high-quality reference genome allowed them to get a much more precise estimate of how much Neanderthal DNA people with ancestry outside of Africa carry today.
And the number was?
Around 1.5% to 2.1%. That refined previous estimates. But here's a really key finding.
Yes.
The Neanderthal DNA found in present-day non-Africans seems to be slightly more closely related to the Neanderthal DNA from Denisova Cave in the Caucasus than it is to the Altai Neanderthal from Siberia, or even the Vindija Neanderthals from Croatia.
What does that imply?
It suggests that the main interbreeding events between Neanderthals and the ancestors of modern non-Africans probably happened after different Neanderthal populations had already started to diverge genetically from each other. The group that mixed with our ancestors might have been genetically closer to those Caucasus Neanderthals.
Yeah, that's fascinating. So it wasn't just one generic Neanderthal population mixing with one modern human population.
Yeah.
The timing and location mattered.
Exactly. They estimated the split time between the specific Neanderthal population that contributed genes to us and this Altai Neanderthal lineage.
When was that?
Somewhere between 77,000 and 114,000 years ago, which is, quite a bit later than when classic Neanderthal features first appear in the fossil record, maybe 230,000 years ago or even earlier.
So supporting recent interbreeding, not just some kind of ancient shared ancestry.
Right. It strengthens the case for actual admixture events happening relatively recently, likely as modern humans expanded out of Africa.
What about the Denisovans? Did the study clarify their contributions too?
Yes, it provided more evidence for Denisovan gene flow. They confirmed a significant contribution to populations in Oceania, places like Australia, Papua New Guinea, Melanesia, where estimates range from maybe 3 to 6%.
And elsewhere.
They also found traces of Denisovan ancestry, but at much lower levels, around 0.2% in people across mainland Asia and even in the Americas.
The big difference between Oceania and the mainland.
Yeah, it is. And they raised an interesting point. They couldn't completely rule out the possibility that the low level of Denisovan ancestry seen in mainland Asians might actually stem from later migrations of people from Oceania who already carried that Denisovan DNA rather than direct mixing on the mainland itself.
Another layer of complexity. Okay, let's look at figure 5. It talks about archaic population differentiation.
Yes.
What was the key message there?
Figure 5 digs into the genetic diversity within the archaic groups around the time of interbreeding. They compared the genetic distance, essentially.
Oh.
They looked at the Neanderthal DNA segments found in Eurasians today and compared how different those segments were from the Altai Neanderthal genome. Then they did the same for Denisovan DNA segments in Papuans and Australians, comparing them to the Denisovan genome.
And the result?
They found that the Neanderthal segments in Eurasians were generally less diverged from the Altai Neanderthal than the Denisovan segments in Oceanians were from the sequenced Denisovan. It fits with other evidence suggesting that Neanderthals, at least the ones we have genomes from, might have had lower overall genetic diversity, maybe smaller effective population sizes, compared to Denisovans around the times these interbreeding events were happening. Denisovans might have been a larger or perhaps more structured population.
Interesting. Now, we've talked about Neanderthals and Denisovans mixing with modern humans, but did they mix with each other? Figure 6 seems to address that.
Yes, another major finding was clear evidence of gene flow from Neanderthals into Denisovans.
How much?
They estimated a minimum contribution of about 0.5% of the Denisovan genome came from Neanderthals.
And was it from any particular Neanderthal group?
Well, intriguingly, the Denisovan genome shared more derived alleles. Those are the changed versions of genes compared to the common ancestor with the Altai Neanderthal specifically than it did with the other Neanderthal genomes from Vindija or Mizmiskaya.
So the Neanderthals who mixed with Denisovans were likely more closely related to the Neanderthals living in the Altai region.
That's the implication, yes. It suggests the gene flow came from a Neanderthal population genetically closer to the Altai individual whose genome they sequenced.
And the paper highlights specific areas, like the HLA region and the CRISP gene cluster, as having strong signals of this Neanderthal input. Why are those regions significant?
They're biologically really important. The HLA region is absolutely critical for the immune system, recognizing pathogens and so on. The CRISP gene cluster is involved in sperm function and fertilization.
So getting Neanderthal versions of genes in those areas could have actually impacted Denisovan biology.
Potentially, yes. It raises the possibility of adaptive introgression, that these Neanderthal genes might have provided some advantage, perhaps related to immunity in their environment or aspects of reproduction, and so they were favored by natural selection within the Denisovan population. We see similar things hypothesized for archaic genes in modern humans.
Okay, now for perhaps the biggest surprise of the paper. The evidence suggesting gene flow into Denisovans from a completely unknown archaic group. This is figure 7. Tell us about this unexpected twist.
This was, a real shocker. It came from a puzzling observation when comparing Neanderthal and Denisovan genomes to modern African genomes.
What was the observation?
They found that African genomes counterintuitively share slightly more derived alleles with Neanderthals than they do with Denisovans. This was especially true for alleles that are fixed, meaning found in almost everyone in Africa.
Why is that weird? Neanderthals and Denisovans are supposed to be sister groups, both outside Africa.
Exactly. You might expect Africans to be equally distant from both, or maybe even slightly closer to Denisovans if some models are right. Finding they shared more derived alleles with Neanderthals needed an explanation.
And the best explanation involved another group.
After ruling out other possibilities, the most plausible scenario they landed on was gene flow into the Denisovan lineage from a much more ancient, unknown, archaic hominin.
Not Neanderthals, not modern humans. Someone else entirely.
Someone else entirely. A group whose ancestors split off from the lineage leading to Neanderthals, Denisovans, and modern humans a very long time ago.
How long ago?
Their estimates for this divergence were incredibly deep, somewhere between 0.9 million and possibly even 4 million years ago.
Wow, that's way back.
Way back. And they estimated that maybe 0.5% up to potentially 8% of the Denisovan genome might actually originate from this mystery archaic population.
Does this connect to any known fossils, like Homo erectus?
The time frame, especially the older end of the estimate, definitely overlaps with the time Homo erectus was around in Asia. So it's a tantalizing possibility that this unknown group could be related to Homo erectus, though proving that requires more evidence, maybe ancient DNA from an erectus fossil itself, which is extremely challenging.
So our family tree just got even bushier and more interconnected. Amazing. And they did check that the Denisovan DNA in places like New Guinea wasn't actually from this mystery group.
Yes. They specifically confirmed that the unique Denisovan ancestry found in Papuans and Australians today comes from the main Denisovan lineage, not predominantly from this deeper, unknown, archaic source.
Yeah.
The signal matched the sequenced Denisovan genome.
Good to clarify. The paper also briefly mentions differences in gene copy numbers and then builds a catalog of changes unique to modern humans. What were the highlights there?
On the copy number front, they found a few regions duplicated just in the modern human lineage after splitting from the archaic groups. One interesting example was a region containing the BOA2 gene.
What does that do?
It's involved in iron metabolism and possibly immunity. Archaic humans seem to have had just one copy, while most modern humans have multiple copies. Differences in gene dosage like that can sometimes have significant functional effects.
And the catalog of modern human-specific changes. What did they find that makes us, well, us, genetically speaking?
A key aim was to pinpoint genetic changes that became fixed or nearly fixed in modern humans after our lineage diverged from Neanderthals and Denisovans. Despite the millions of years, the list of fixed differences was actually relatively small.
How small?
Around 30,000 single-base substitutions across the whole genome, and about 4,000 small insertions or deletions that were fixed in modern humans but ancestral in the archaic genomes in apes.
And changes that affect proteins.
Even fewer. They identified only 96 amino acid substitutions, affecting 87 proteins, that were fixed in modern humans.
That doesn't sound like a huge number, considering all the differences.
It's not huge, but where those changes occurred is fascinating. They looked at where the genes containing these fixed amino acid changes are expressed in the body.
In any patterns?
Yes, there was a significant enrichment for genes highly expressed in a specific part of the developing brain, the ventricular zone of the neocortex.
That sounds important for brain development.
Extremely important. It's where neural stem cells proliferate. Some of the genes with fixed changes included ones involved in the machinery of cell division, the mitotic spindle like KSC5, KVNEAA, SPAGE5. Another VCAM1 is involved in maintaining those neural stem cells.
Suggesting genetic changes related to brain development might be key differences.
It certainly points in that direction. They also identified 63 genomic regions showing signs of positive selection, specifically in modern humans. These regions contained over 2,000 substitutions and some insertions deletions, again including genes implicated in neural stem cell function like RB1-CC1.
So, providing crucial clues about the genetic underpinnings of uniquely modern human traits, especially related to our brains.
Exactly. It's a starting point for functional studies to figure out what these specific changes actually do.
Okay, let's try to wrap this all together. Looking back at the whole study, the discussion section, figure 8, what are the really big picture takeaways?
Well, the overarching theme is complexity. The evidence points to a late Pleistocene world, not of isolated groups, but of at least, 3, maybe 4, even 5 distinct hominin lineages interacting and interbreeding at different times and places.
Neanderthals, Denisovans, modern humans, that unknown archaic group.
Right, but interestingly, despite these multiple instances of contact, the overall amount of gene flow between the groups seems to have been relatively low, a few percent here, half a percent there.
So contact happened, but maybe it wasn't constant or widespread mixing.
Perhaps. The study powerfully confirmed that the Neanderthal ancestry in non-Africans today is definitely the result of relatively recent interbreeding, not just deep shared ancestry. And that discovery of gene flow into Denisovans from the unknown archaic group, that just opens up a whole new chapter.
And the significance of this specific Altai genome.
It became the high-quality reference point. It really moved ancient genomics into a new era, allowing for much finer detail in studying human origins, how we adapted, even our susceptibility to diseases linked to this ancient DNA.
Okay, as we finish this deep dive, what are the core insights you think people should really take away from this research?
I'd say 3 things. First, our evolution wasn't linear. It was a network, a web involving interbreeding between different kinds of ancient humans. Forget the simple trees.
Right.
Second, that startling evidence of inbreeding in the Altai Neanderthal gives us this incredibly intimate glimpse into the possible social structure and demographic challenges faced by at least some ancient populations. Makes them more real.
And third,
Just the power of having a high-quality ancient genome. It became this invaluable resource, a benchmark for understanding what makes us genetically unique today, and how echoes of that archaic past still resonate within our own DNA.
what was the biggest aha moment for you in reading or rereading this paper?
Honestly, it's still that inbreeding finding. The sheer fact that they could determine her parents were likely half-siblings. It's just such a specific, personal detail from over 50,000 years ago. It collapses time in a way and forces you to think about these individuals not just as data points, but as members of families and small communities.
And why should we care about all this ancient history today? What's the relevance or application?
Oh, it's hugely relevant. Understanding our genetic relationship with Neanderthals and Denisovans is key to understanding what makes us tick biologically. It helps us identify the genetic changes that might underpin unique modern human traits, like aspects of our cognition or behavior.
And the health connection.
Yes, absolutely. We now know that some of the DNA we inherited from these archaic groups influences our immune systems, our metabolism, our susceptibility to various diseases, both positively and negatively. So studying these ancient genomes directly informs modern human health research. This 2013 paper really laid critical groundwork for so much of that subsequent work.
Okay, finally then, leave us with a provocative thought. Something this research makes you wonder about, something for us to chew on.
Well, given all this evidence for repeated interbreeding Neanderthals and humans, Neanderthals and Denisovans, Denisovans and humans, Denisovans, and this unknown group, it really forces you to question our neat definition of species when we talk about these late Pleistocene hominins.
Were they truly separate species if they could interbreed successfully, even if only occasionally?
Exactly. Maybe the biological reality was less about distinct reproductively isolated boxes and more like a fluid network of populations across the landscape capable of exchanging genes when they met. How should we conceptualize that? This high-quality genome didn't just give answers. It opened up fundamental questions about boundaries, identity, and the very nature of human evolution that we're still grappling with today.